but not the nuclear membrane. So you end up with some volume of soup, really, free cell nuclei just sloshing around in that volume, and you choose what that volume is going to be. You can just round it up to a number that you can read on a graduated cylinder. You stain that, so you mix that, so now you have a perfectly homogeneous, the proper word is actually isotropic suspension of nuclei. And if you stain that with DAPI, you can take any sample now, and that sample should be representative of the whole. And you can ascertain that that is the case by taking those samples to the microscope and just counting them quickly with a helicytometer. So this is what free nuclei look like. It takes 10 to 15 minutes to count 4 samples under the microscope directly and get your number. I always get asked, why don't you just let fax machines do the counts for you? I'm like, I can beat the fax machine anytime. All it takes me is 4 little samples. I go to the microscope and I have it in 10 minutes. I don't have to reserve machine time. I don't have to filter my sample. I don't have to calibrate anything. It's really fast. And in 2 more hours, you do staining with NeNa of the free nuclei, and this is what it looks like. So you see there's essentially 0 background here. It's very easy to tell what cells are neurons. And therefore, you have, in not more than a few hours, you know how many cells your original structure had. You know what fraction of them were, in this case, neurons. By subtraction, you know how many non-neuronal cells, glial cells, and vasculature you had. And now that we have other nuclear markers that can identify different cell types, we can use that as well. It's something that that's something that we have now. I'm not going to show you in the talk, but we can look at microglial cells and astrocytes and oligodendrocytes in separate. And what we find here for total non-neuronal cells applies to each and every one of those cell types. So, what then? You want to understand diversity, you have to be able to look at diversity. So, I've been able to do that thanks to a very, very large number of collaborators, and that involved doing weird things like sending an email to somebody I had never met, Paul Menger, but his name kept popping up when I talked to people about how can I find brains of different species? Do you know anybody that works with different brains? The answer used to be Paul Menger, and I came across a paper where he described how to perfuse elephants in the wild and collect their brain for histology. Very well conserved, everything perfectly fine. So I figured by this point we had just published a paper on how many neurons human brains are made of. So I figured I'm going to send him an email and I'm just going to ask, because the worst that can happen is if I don't ask, I have no elephant brain, right? So I sent him an email trying to make a good case, but really what the email said was, hello stranger, I see you have elephant brains. I like turning them into soup. Could I have a hemisphere, please? Right? It worked out because Paul, once you meet him, you realize that he is very fond of weird, unorthodox ideas. So he replied nicely saying, whole hemisphere is a bit too much, but I can give you a few pieces. Is that all right? I'm like, well, thank you so much, but actually no. If we just get pieces, we're not going to be able to go past what's been done in the literature, which was just looking at densities. The problem with looking at densities is that if you use densities to obtain estimates of numbers of neurons by simply multiplying the density by the size of the brain, you get a result that is mathematically dependent on that huge, very variable brain mass. So you get mathematical contamination, and you end up with something that looks very close to a linear function that doesn't necessarily have to be a linear function, right? So you actually need proper counts, direct counts of neurons. So I wrote right back to him, made my case on somewhat lengthy email, and to my surprise, I got an email not much longer after that saying, okay, I like your sales pitch. I'll send you a brain hemisphere. And they're like, no, 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 no. Don't send it in the mail. Please do not put an elephant hemisphere in the mail. I will come get it. I was in Brazil. I'm not going to get started telling you about all the ordeals that bureaucracy and customs and everything is like. Let me just summarize it this way. It was cheaper and faster to buy a ticket to fly from Rio to South Africa and come back than to go through all of that. So anyway, it turns out that Paul liked the idea very much, and I came back with dozens of brains, not just one, and that is essentially the reason why we've been able to start mapping diversity in brain evolution, and we were able to figure out a number of things. Let me start then with something that apparently really does not change. So if you look at the mass of a structure as a function of how many other cells, so at this point now I can really say that these are essentially glial cells across species. Each dot there is one part of the brain of one different species. And you see that over 6 orders of magnitude each way, you pretty much have the same thing going on. You have essentially the same relationship here that is very, very close to linearity. What that means is that if you look at the density of the glial cells in the tissue across all these species, and this is all different brain structures mixed together, you see that there is some variation, but there's no systematic variation here. Essentially what this indicates is that the density of glial cells is very much the same across structures and species, which is not at all what you find when you look at the density of neurons across structures. This is the cerebellum, so you see that there's a much higher density of neurons in the cerebellum than in the cortex, than in the brainstem. And also you see that there's a very steep slope here. Neurons in the cortex and even more in the brainstem, they apparently do become much larger on average as you gain neurons. The consequence is that we were able to show that it's not that the number of glial cells per neuron increases with the size of the brain or of the structure. That's not true because these densities are extremely variable. But you take variable neuronal densities with fairly unvariable glial cell densities, and what you get is essentially a glia to neuron ratio that scales uniformly across all these species, and even birds. The black here is birds, as neurons become bigger. So as the density decreases, you have larger ratios. You have more glial cells per neuron, right? Doesn't matter what species, doesn't matter what part of the brain. If you tell me what is the density of the neurons, I can tell you how many glial cells you are likely to find per neuron. So neurons then, there is variation. This is how the mass of the cerebral cortex, both gray and white matter, vary across species as a function of the number of neurons in the cortex. So this is all cerebral cortex. Each dot here is one different species. Color coding is what I had in that evolutionary, in that tree. So red are primates. Everybody else are these different colors here. So you see that there seems to be one relationship that applies to one relationship that applies between the size of the cerebral cortex and how many neurons it's made of if you're not a primate. But if you are a primate, there is a different relationship. But very important thing, humans, us here, we're right on the line. We have exactly the same relationship between the mass of the cerebral cortex and how many neurons it has that you would expect for a generic primate, right? And the difference here has to do with what happens to neuronal density as cortices gain more neurons. So what we find for the non-primates is very similar to what people had been expecting, that the more neurons you have, the lower the density, and remember that in the context of glial cell densities that are essentially unchanging, this has to mean mathematically that the more the neurons you have, the larger they are on average. Except if you're a primate. If you're a primate, the density, there's some variation, but it's nothing as dramatic as you find across other species. There might be a very shallow slope here, but it's very, very shallow. Now, the interesting thing is that even though these are all modern species, they exist today, because there's this nice clustering, there's this nice single relationship here to everyone that's not a primate, and because this is, to our knowledge, the best evolutionary relationship that we can attribute to them, it really looks like whatever scaling relationship this is here that applies to everyone that's not a primate, so all these here in green, it's the most parsimonious thing to say in terms of evolution is that this scaling relationship, rather than appearing again and again and again and again and again and again independently, it probably was the way that brains were put together when the first mammals appeared, right? Now the useful thing is that from the fossil record, we know that the first mammals were very small, with very small brains, with very small cortices, and if they had very small cortices, then they must have had very few brains that were also very few neurons that were also very small neurons, right? So we can infer from the modern day data that mammals started out somewhere around here. And as numbers of neurons increased in some species, those neurons somehow also became larger. That was, having neurons become larger was the thing that followed having more neurons as well. And then, at some point, some 50-something million years ago, when a group of animals diverged away from that, it must have been with something, some change that uncoupled having more neurons from having the average neuron become larger. And there you have primates, a group where more neurons in the cortex, you have more neurons in the cortex, but the density doesn't really change, right? So you have essentially these two modes of building cortical tissue, building cortical volume. You have the primate and the non-primate group. Now, here's the thing. This is volume. Volume can actually be spread laterally in different ways. And the way I like to put this is with our jam and cracker model of how you spread neurons in the cortex. I will try to convince an editor to let me use these terms. So here's the idea. You have a certain volume of cortex, that's your jam, that is made of a predictable number of neurons. That's the fruit in your jam, right? So remember that if you're not a primate, there's always a constant number of neurons in a certain volume of jam, depending on the size of your cortex. That's predictable. But that volume can be spread very thick over a small surface area, or it could, in principle, be spread very, very thin if it forms a larger surface area, right? So even though non-primate cortices have a volume of cortex that is built the same way as a function of the number of neurons, so depending on how large the average neuron is, that volume, you can get to the same volume if you have a cortex that's like this or like this, right? How does that apply to our, let me go back to where we were, how does that apply to all these cortices? So from here, you might still think, well, there's cortical diversity is restricted to just two modes. You have the primate way of building cortices, and you have everybody else's. That's true in terms of volume, but it turns out that in terms of surface distribution, you have very, very different relationships. Each clade seems to do its own thing. This is how the thickness of the cortex varies as a function of the surface area of the cortex. And you see that the relationship is entirely different depending on whether you are a rodent here in green, whether you're a marsupial here in orange, whether you are an arthodactyl here in pink, or whether you are a primate. So the slope is completely different in red. So you have these two different ways of putting a cortex together that also translate in how the number of neurons that you get in the cortex are actually distributed, now not in the volume, but across the surface and the thickness and the surface of the cortex. So this is how the thickness scales with the number of neurons, and you see that each clade does its own thing. And here's how the surface of the cortex scales as a function of the number of neurons, which also does its own thing. Maybe it doesn't seem as dramatic as when you're looking at thickness, but here is what that translates into. Remember that consensus that all brains or all cortices have a constant number of neurons per surface area? Well, now we have estimates, direct estimates of total numbers of neurons in cortices, and we know the surface area of each of these cortices. So you divide one thing by the other, and you get the average number of neurons underneath a surface area in the cortex. And you can, so this is how many neurons you have per surface area. Remember, the prediction was this has to be constant. This is constant, right? In marsupials, maybe it is, but in rodents, you clearly have fewer and fewer neurons underneath the surface area. In arthodactyls, even more so. But in primates, not only you don't have that, you also have a much larger number of neurons per surface area. Now, notice that if you do count neurons in this group of species around here, you might think that they have pretty much the same number, right? That is why, if you want to look at diversity, you really have to look at a diverse group of species. The usual mouse and rat and marmoset and monkey is not gonna do. But anyway, so we can put this together then, and we can go back to those initial predictions. It's essentially what we have is that there's none of this. There are no universal relationships here. Each cortex is built in a different way in a different clade. There is also no constant number of neurons per surface area. And what about this? Is there a universal relationship between how folded the cortex is and how many neurons it has? Meaning, is the only way to get more neurons in the cortex having that cortex fold? Or do you necessarily get a more folded cortex as you gain more neurons? Well, now that we have data for all these different species, we could look at that. So this is, just to go back, this is what you would typically find in the literature, simply because nobody had numbers of neurons to look at, and everybody assumed that brain mass was going to be a good approximation of that across all the different species. So what you have here is, remember, a folding index of 1 means your total surface area is the same as your exposed surface area, meaning you have a cortex that is not folded at all, right? So you see here that the smallest brains are never folded. Then there seems to be some critical point after which the larger the brain is, the more folded it tends to be. Except that cetaceans, whales, dolphins seem to have much more folded brains than you would expect for their brain mass. And you have these outliers. Humans here seem to be below the expected. And here you have a manatee. That's the manatee's cortex, shown to scale with the regular cow cortex. You see it's a sizable brain. It's about this big, and it certainly looks like it's missing folds, right? It is apparently lissencephalic. So that's a big exception there. Now, remember that the expectation here would be that this degree of folding should follow universally, homogeneously, increasing numbers of neurons, right? How does that look to you? So in each, remember, each color is a different clade. In each clade, the more neurons you have, the more folded the cortex is, right? But Is it universal across everybody? Essentially what you have here is that a pig cortex is exactly as folded as a baboon cortex, and yet the baboon has 10 times as many neurons in the cortex as the pig has. And also, we have more neurons, we have 3 times as many neurons in the cortex as the African elephant does, despite the fact that the African elephant brain is much larger than ours. That, by the way, is what seems to, let's say, single out humans amongst other species because of the different way in which primate brains are built. Because we're primates and because we have the largest brain amongst primates, we have the largest number of neurons in the cerebral cortex, even compared to other non-primate cortices that are much bigger than ours. So the one variable that most obviously sets humans apart from every other animal species is simply the absolute number of neurons that you have in the cortex. So if that's the case, then you would expect the human cortex to be the most folded of them all, but it's not. So the elephant cortex is about twice as folded as the human cortex, even though it has only 1/3 as many neurons, right? So it can't be, folding cannot be a simple, direct function of how many neurons you have in the cortex, right? It also appears not to be the only way to get more neurons into a cortex. Otherwise, our cortex could never be as little folded as it is compared to an elephant's, right? It turns out that if you have a physicist friend who at that point had already gotten his PhD in topology of the universe and asked, could I please do a postdoc in your lab? And you say, sure, of course. I would very much like to have a physicist working in my lab. So this was Bruno Motta, and we had this ongoing game of just tossing questions at each other. I learned to be very careful about when I would toss a new question at him because he would invariably stop everything else that he was doing and just immerse himself in that new problem. So I learned to just keep my mouth shut at problems, sometimes. So I had this, and I also looked at the data in a different way. I don't think I have that here. It turns out that the closest we could get to a universal relationship was with just absolute surface area of the cortex. But it still didn't look quite right because you had something like this for the lissencephalic species and something like that for the droencephalic species. But still, the droencephalic species fit much closer together with no enormous exceptions. I showed that to him. We talked a bit about how cortices grow and how cortices fold and how they grow under tension, and his physicist mind kicked in, and he came back a couple of weeks later with sheets and sheets of papers with equations scribbled on that all boiled down to an equation that I could understand. And essentially the equation is the one that predicts the relationship between the total surface area, the exposed surface area of the cortex, and the thickness of that cortex. That gives you a certain volume of gray area and white matter underneath that is the most energetically favorable, the most stable. Given that, you have a cortex that develops under tension, under pressure from the CSF, under pressure from the cells pushing against each other, right? So the equation is simply that of a product of total surface area, the square root of thickness of the cortex, and that should scale as one single universal function of the exposed surface area of the cortex. And lo and behold, you get this. You can imagine the commotion in the lab and all the jumping, and that's what data tables are for. And so you have all the data so that you can come up with a hypothesis. You can test your hypothesis by just plug in some numbers into a graph, and you get this. And it's awesome, because finally, for the first time ever, you have something that makes sense for every single species. Remember, there is a difference between saying this works beautifully and this is what happens in nature, right? But still, what we have here is an R square of 0.998. That means that 99.8% of the variation in this product here is explained by variation in the exposed surface area, or vice versa. You had a question?
Yes, I have a question.
Yeah.
So, in your whole research,
Can you speak louder?
okay, so in your whole research, if you consider the longevity of different species,
I'll come back to that in a little bit. Just bear with me. So what we have here is one universal relationship that is derived from first principles, from pure physics, that seems to describe 99.8% of the variation in the degree of folding of the cortex, depending essentially on how large the surface area is and how thick that cortex is. So what we have proposed is that cortical folding is driven by pure physics, by simple energetics of a structure, happens to be biological, but it's still a structure that has a certain surface area and a certain thickness. And folding occurs as the effective free energy of that growing volume with a self-avoiding surface is minimized. So at each point in development, that growing volume settles in the conformation that is the most energetically favorable. So it will just do that naturally. So this, just for comparison, this is what you get when you apply the equation to just the species we have, and that's us again, exactly on the Y. Now the interesting thing is that although the physics seems to be the same, regardless of how many neurons compose those cortices, you can look at all these cortices and still tell this is a primate, this is a carnivorous, this is a marsupial, this is a rodent, from the spatial distribution of those folds. So essentially, what you have here is a clade-specific pattern. So this is biology. That applies to something that no matter where the first folds form, and we're working on figuring out what is that related to. Is it initial differences in the connectivity of the cortex that would just set heterogeneities in the distribution of forces, of tensions in that growing cortex? But whatever they are, they will always lead to a cortex that is predictably folded to a certain degree. So let me recap here before I go into the part of what doesn't matter. What we have here is that you do have cortical diversity that stems from changes in the average size of the neurons, and that varies between primates and non-primates. We do have clade-specific diversity in the lateral spreading of neurons. You can have a certain number of neurons form, but then either cluster vertically or else become spread radially. And so here you have this clade-specific spreading of neurons, and that turns out to also be described by a very simple model that we're working on. And you also have this diversity in the spatial patterning of the cortical folding, but still, all of this happens along with other things that do not change even in the face of so much evolutionary diversity. And one of those things is that the average size of the non-neuronal cells seems to be very constant. Now, remember that the word evolution essentially means change over geological time. So whenever you find something, some feature that doesn't change over geological time, that means that what you're looking at is either subject to, it's either just pure physics, and that seems to be the case with the degree of folding of the cortex, or else is something that is under a very strict biological limitation. And this is something else that we're working on now, we're figuring out what are these biological limitations. What the best guess is that if the size of the glial cells, and let's talk about astrocytes for a little bit. If the size of the astrocyte is not very